Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing entrance exam question paper for GAT B, which was conducted in the year 2023. This is part B questions. Part A I have already uploaded the video. You can go through that. Then coming to the first question. Which of the following is not a preparative purification method for recovering protein product after fermentation? So it is thin layer chromatography. They have asked after fermentation, thin layer chromatography is not used. It is only for qualitative analysis, thin layer chromatography will be used, but not for industrial purification purposes. Then next one, retrotransposon category and example. So retrotransposon, they move by the action of RNA intermediates. They produce RNA transcript and they use reverse transcriptase enzyme to convert RNA to DNA, which is then inserted into the target site. There are two major types of retrotransposons, that is LTR and non-LTR. So LTR, they are characterized by the presence of long terminal repeats on both the ends, whereas non-LTRs, they lack this repeats. Then there is autonomous and non-autonomous transposon. Autonomous can move on their own, while non-autonomous elements require presence of other transposon elements in order to move. That is non-autonomous, they may lack the gene for reverse transcriptase that is needed for transposition, and they must borrow this reverse transcriptase from another transposon. They are non-autonomous. So now we will see they have given examples of autonomous, non-autonomous, LTR and non-LTR one. You have to match. So LTR and autonomous A3 copia, non-LTR and autonomous I factor B4, LTR and non-autonomous C1 dashing, and non-LTR and non-autonomous D2, that is ALU. So the answer here is option 2, that is A3, B4, C1 and D2. Then next one, which is not a part of mandatory design consideration of a rotating biological contactor system. So in a rotating biological contactor, it is used in the secondary treatment of wastewater. And in this process, the wastewater is allowed to come in contact with the biological film to remove the pollutants before this water is being discharged to rivers or oceans. So they have asked which is not a part of mandatory design considerations of a rotating biological contactor. It is rotation speed. Answer is option 4. Then next one, which of the following conditions would in general be more conducive for the production of roots from callus tissues of plants under in vitro conditions? So you need higher auxin ratio, because auxin is needed for root initiation in tissue culture plants. So higher auxin ratio and lesser cytokinin ratio will be required. So the answer is option 2. BCG vaccine is an example of, so it is an example of live attenuated vaccine. Answer is option 1. Then next one, which of the following antibody is used by immune complexes for complement activation? It is IgM and IgG, which are used for complement activation. So they have given IgM only here, IgG is not given. So answer is option 2. First clinical use of gene therapy was done for treatment of, so it was done for the treatment of adenosine deaminase deficiency. Answer is option 2. Which of the following is true for the free energy change of a reaction delta G catalyzed by an enzyme? A reaction take place spontaneously only if delta G is negative. That itself is correct. Next one. If a system at equilibrium, there is a net change in the concentration of products and reactants and delta G is positive. So in equilibrium, the concentration of products and reactants will not change. So option 2 is, statement 2 is not correct. The delta G of a reaction is dependent on the path of the transformation. It is independent of the path of the transformation. Then fourth one, the delta G provides information about the rate of a reaction. No, it tells about the feasibility of the reaction, not about the rate of the reaction. So option 1 is the answer. Then which of the following bioprocesses would yield maximum number of ATP per gram of substrate consumed? So the one with lesser molecular weight. If you see methanol, it has a molecular weight of 32 and acetate is having molecular weight of 44 and glucose is with 180. So it is having higher molecular weight. So methanol is the answer. Aerobic catabolism of methanol. Because smaller molecular weight, more energy is released. And moreover, methanol is the most reduced form out of glucose and acetate. So more reduced form means it has more energy. So aerobic catabolism of methanol is the answer. Then next one. The purity of a plasmid DNA preparation is determined by analyzing the ratio A260 by A280. In a pure DNA preparation, the ratio will be approximately 1.8, or I can say it is close to 2 as per the options given here. If it is equal to 2 or approximately equal to 2, then it is pure RNA preparation. So the answer here is option 1. Then next one. Which of the following is an RNA virus? So dengue is single-stranded positive sense RNA virus. That itself is the answer. Smallpox virus is double-stranded DNA virus. HPV or human papilloma virus, it is double-stranded DNA virus. And hepatitis B virus is small DNA virus. So the answer here is dengue virus. The next one. Which of the following is not a PCR based method? So out of these, restriction fragment length polymorphism or RFLP is not a PCR method. It involves the use of restriction enzyme and southern blot. So whereas the other three are PCR based methods. The next one. Which of the following is normally used to determine the frequency of interferon gamma producing cells in a blood sample? So it is ELISpot that is used. ELISpot, it is a method for quantification of the number of analyte secreting cells. Analyte secreting cells means it can be your cytokines or immunoglobulins. So they have asked here for interferon gamma producing cells. So it is ELISpot. So they have asked for interferon secreting cells. So that can be determined by ELISpot method. Then next one. Which of the following is the least abundant immunoglobulin class in a normal adult? So it is IgE. It is the least abundant immunoglobulin class. In which stage of HIV infection does one usually show symptoms of AIDS? So when HIV damages a large number of helper T lymphocytes, because HIV infects mainly the CD4 plus cells or helper T lymphocytes. So when these cells are being damaged, they usually show the symptoms of AIDS. So answer is option 3. Then next one. Information about the isoelectric point will be useful in the purification of a protein by which of the following chromatography techniques? So it is ion exchange chromatography, chromatography. Because isoelectric point is the pH at which protein has no net charge. So if you want to select a cation exchanger or an anion exchanger for the purification of your protein, you have to know the isoelectric point, because if the isoelectric point is above, if if the buffer pH is above its isoelectric point of the protein, it will be negatively charged and will bind to an anion exchanger. Now the buffer pH is below its pi, the protein is positively charged and will bind to a cation exchanger. So the answer here will be ion exchange chromatography. Answer is option 2. Then next one. Intine, the inner wall of a pollen grain is made up of. So it is made up of cellulose and pectin. Sporopollenin is present on the exine, not on the intine. Then next one. IgM. IgM is a pentamer and is involved in complement activation. They had asked this question just for 6-7 questions before. For regarding IgM, that time I told you IgG is also involved in complement activation. So A4. Then IgA. IgA is a monomer, dimer, trimer and mucosal immunity. Then IgD is a naive B cell antigen receptor, and IgE is a monomer and mast cell activation. So the answer here is option 4, A4, B3, C2 and D1. Then next one. Which of the following can be used for cell line authentication? So for cell line STR analysis is universally accepted method for human cell line authentication. And the procedure, it requires PCR. So answer here is option 3, that is PCR. EST stand for expressed sequence tag. Answer is option 2. Then log P is a measure of drug likeliness in Lipinski's rule of 5. P denotes. So Lipinski rule of 5 help in distinguishing between drug-like and non-drug-like molecules. So the P here, it represents a partition coefficient. Answer is option 1. Then next one. The equilibrium sedimentation experiments performed by Michelson and Stahl in 1957 demonstrated that DNA replication takes place in a semi-conservative fashion. Answer is option 1. Then the next one. Theoretical RQ, respiratory quotient for the complete oxidation of pyruvic acid will be. So respiratory quotient is the ratio of carbon dioxide produced to oxygen consumed. So when pyruvate is cycled through TCA cycle, it produces 3 molecules of carbon dioxide and consumes 2.5 oxygen molecules. So respiratory quotient will be carbon dioxide produced, that is 3, divided by oxygen consumed, that is 2.5, and it will give you 1.2 as the answer. Answer is option 1. Next one. Which of the following biochemical techniques does not use antibody? So isoelectric focusing does not use antibody. Here the analytes are separated using the principle of isoelectric point. Then next one. Which of these viruses make use of RNA-dependent DNA polymerase during replication? RNA-dependent DNA polymerase is reverse transcriptase. So which viruses use reverse transcriptase? It is HIV. Answer is option 1. Then next one. Which of the following is a bacteria known for high yield of ethanol production? So it is not Saccharomyces cerevisiae. Yeast is a fungi. It is not bacteria. So they have asked bacteria over here. It is Simonomonas mobilis. Answer is option 3. Then next one. Which of the following is not an example of bioremediation? So, depletion of ozone by Aspergillus niger. Answer is option 4. Then next one. Which of the following statement is not true about the seed culture of orchids? They grow on poor salt poor medium as mineral requirement is low. Protocomb like structure is formed from the embryo. That is also correct. 1 is correct, 2 is correct. Seeds are very small and contain little or no reserve food. That is also correct. In vitro germination and development is very slow in orchids. That is not correct. In natural conditions, the seeds of orchid, they take long time to germinate. But in vitro propagation of orchids through seeds can produce large number of orchids in reasonably short time. So the answer here is option 4. Then next one. Which of the following is best suited to express large amount of glycosylated protein? So it is baculovirus. Answer is option 3. Then next one. Which of the following is correct about the endoplasmic reticulum? The smooth endoplasmic reticulum is involved in the glycosylating the proteins. It is not the smooth, it is the rough endoplasmic reticulum. The rough endoplasmic reticulum is involved in the synthesis of proteins that will either be inserted into cellular membranes or be secreted from the cell. That is correct. The lumen of rough endoplasmic reticulum is highly reducing in nature. It is not reducing, it is oxidizing in nature. The smooth endoplasmic reticulum synthesizes globular proteins and a rough endoplasmic reticulum synthesizes fibrillar proteins. Smooth endoplasmic reticulum is not involved in protein synthesis. So statement 4 is not correct. The next one. Which of the following amino acid contribute to the sulfur content in a protein? Methionine and cysteine. They both contribute to sulfur content in the protein. So if you see the structure of methionine and cysteine, they will have a sulfur in their structure. So the answer here is methionine. In a reaction, if P product is at a lower internal energy level than S substrate, then the reaction is exothermic. Answer is option 2. Then, cytokine released in response to viral infection is termed as. Cytokine released in response to viral infection is interferon. Answer is option 2. A humanized murine antibody is one in which. So in monoclonal antibodies, in the mouse monoclonal antibody produced will be considered as an antigen by the human body and it will result in the rejection reaction being taking place. So to avoid this, you use a humanized antibody. So humanized antibody, it contains the human constant domains of the antibody and mouse variable domains, monoclonal mouse variable domains. So they will not be rejected by the human body because it is human constant domains are present. So that is called a humanized antibody. So here it will be complementarity determining regions of the are from the mouse and other regions from the humans. These what they mean by other region is the constant regions of the human antibody. So the answer here is option 4. Then next one. Which of the following is a dominant genetic trait in humans? So alkaptonuria, it is autosomal recessive. Sickle cell anemia also autosomal recessive. Huntington disease is autosomal dominant. And phenylketonuria is autosomal recessive. So the answer here is option 3. Then which is the most abundant protein in the entire biosphere? That one which is present in the plants. So that is ribulose biphosphate carboxylase, oxygenase or rubisco. Answer is option 4. Then next one. Which of the following is desirable in a primer for PCR? So should be more than 50 nucleotides in length. The primer length should be between 18 and 24 base pairs, not more than 50 nucleotides in length. Secondary structures such as hairpins. Hairpins and secondary structures shouldn't be produced by the primers used for PCR. Sequences with stretches of identical single nucleotides. There shouldn't be sequences with stretches of identical single nucleotides. Then minimal complementarity between reverse and forward primers. That is desirable for PCR primers, because if complementarity between forward and reverse primer is not there, then primer dimer will not be formed and the primers will bind to the target DNA. So the answer here is option 4. Then next one. Which of the following processes involves the formation of nitrate and ammonia? Ammonification. It produces ammonia from breakdown of organic nitrogen. Denitrification. Nitrates or nitrites are broken down by liberating free nitrogen into the atmosphere. Nitrification is ammonia is sequentially oxidized to nitrite and nitrate. And nitrogen fixation is the process in which nitrogen gas is converted into a usable form for plants and other microbes. Nitrogen gas is converted into ammonia and other related nitrogenous compounds. So the answer they have asked for the process involves formation of nitrate from ammonia is nitrification. Answer is option 3. Then neurotransmitters can be either excitatory or inhibitory depending on where they are released, what receptors they bind to, and the ionic condition that they encounter. Which of the following is a group of excitatory neurotransmitters? So an excitatory neurotransmitter, it promotes the formation of an action potential in the receiving neuron, while an inhibitory transmitter, it prevents the formation of an action potential. So out of these, GABA and glycine, they are inhibitory neurotransmitters, whereas option 3, that is glutamate, acetylcholine and serotonin, they are excitatory neurotransmitters. The next one. A gene whose expression helps to identify transformed cells is known as. So it is called as selectable marker gene. Answer is option 4. Then which of the following statements are not true for photochemical smoke? So photochemical, it is an oxidizing smoke. Its main component is sulfur dioxide. No. Photochemical smoke, it contains high concentration of ozone HNO3, whereas the classical smoke, it contains high concentration of sulfur dioxide, not photochemical smoke. It occurs in warm and dry climate. It is formed due to the burning of fossil fuels. So the answer here is option 2. Its main component is sulfur dioxide. Then trihydroxypropane is commonly known as glycerol. Answer is option 3. Anaerobic digestion proceeds in 4 steps. Which of the following represent the correct order of these steps? So in anaerobic digestion, the microorganism break down the biodegradable material in the absence of oxygen. And the processes that take place include hydrolysis, acidogenesis, acetogenesis and methanogenesis. Then next one. The distance between each turn in the helical strand of DNA is, so it is 34 Armstrong. Answer is option 2. Then next one. Given below are two statements. Statement 1. The tDNA regions of a naturally occurring TI plasmid of agrobacterian tumefacients contain genes for biosynthesis of plant hormones. Statement 2. The virulence genes of agrobacterian tumefacients are placed on a helper plasmid during development of a binary vector system for plant transformation. In the light of the above statement, choose the correct answer from the options given below. Both statements 1 and statement 2 are true. Both the statements are correct. So the answer here is option 1. Then which of the following is a heteropolysaccharide? A polysaccharide that which contain different types of monosaccharides is a heteropolysaccharide. Now, chitin contains N-acetyl glucosamine residues in beta linkages. Dextran, it is a homopolysaccharide and is composed of alpha-1,6 linked D-glucopyranose residues with alpha-1,3 branched linkages. I am telling this because next year if they ask regarding what is the dextran homopolysaccharide composed of, you should be able to answer. For that only I am telling about the constituents of each. Then agarose. Agarose is made of alternating beta D galactose and 3,6 anhydro L galactose units of agarobioes. So agarose is a heteropolysaccharide. Glycogen, it is a homopolysaccharide of alpha D glucose units. Then next one, which antibody is involved in type 1 hypersensitivity reaction? It is IgE. Answer is option 3. Which of the following is generally not a step in PCR? It is ligation. Denaturation, elongation, annealing are steps in PCR, whereas ligation, joining two ends of the DNA, it is not a step in PCR. Then next one, to combat a severe viral infection, what kind of immune response would be elicited? So both humoral and cell mediated response will be elicited. It is a severe viral infection. Then next one, the UN Conference on Environment and Development is popularly known as, so it is Rio de Janeiro Earth Summit, Rio Summit, Rio Conference and Earth Summit. So they have given Earth Summit here. So answer will be option 4, Earth Summit. Which of the following exhibits maximum mobility in an agarose gel? It is supercoiled plasmid DNA because it is more compact conformation. So it moves through the gel very fast. So the answer here is option 2. The next one. Charles Richet was awarded Nobel Prize in Physiology or Medicine for his seminal discovery of hypersensitivity. Answer is option 2. Then next one. The structural gene LAC-A in the lac operon is responsible for. So LAC-A codes for transacetylase and enzyme which converts lactose into its active form, that is allolactose. So the answer here is option 3. Then next one. Choose the correct answer from the options given below, that is the best possible match of various components of plant tissue culture media and their role. So list 1 is the components of plant tissue culture media and 2 is the role of each. So nitrogen. Nitrogen is a macronutrient A2, sucrose is a carbon source B1, manganese is a micronutrient C4, myoinositol is an organic supplement D3. So the answer will be option 2, that is A2, B1, C4, D3. Then next one, match list 1 with list 2. Ampicillin. Ampicillin, it inhibits cell wall synthesis by disrupting peptidoglycan cross-linking. A3. Kanamycin inhibits translation by binding to 30S ribosomal subunit. Hygromycin B, C4, inhibits translation by interfering with ribosomal translocation. D1, chloramphenicol, inhibits translation by binding to 50S ribosomal subunit. So the answer here is option 1, A3, B2, C4 and D1. Then next one. Which of the following would in general not be a preferred choice for transgenic plants? Transgenic plants with higher level of transgene expression. No harm, you can go for it. Transgenic plants with single copy insertion of the transgene. Yes. Transgenic plants without any pleiotropic effect of the transgene. Yes. Transgenic plants with integration of multiple copies of the transgene. No. So that is not a preferred choice. So the answer here is option 4. Then next one. Most bacteria require vitamins as growth factors. Answer is option 1. Symptoms of an allergy attack can be prevented by. So blocking the attachment of IgE antibodies to the mast cell. Because once they attach to mast cell, they will release the histamines, which will cause an allergic attack. So if you block the attachment of IgE antibodies to the mast cells, then you can prevent the symptoms of allergic attack. Then next one. Which of the following is not a multiple sequence alignment program? So it is AMBER, AMBER. It is assisted model building with energy refinement. Allows users to perform molecular dynamic simulations on biological systems. So it is not a multiple sequence alignment program. The next one. The genome of hepatitis B, a DNA virus, is unlikely to code for. So it is unlikely to code for RNA-dependent DNA polymerase. Then next one. Which of the following could be the recognition site of a type 2 restriction endonuclease? It should be a palindromic sequence. Then it will be recognized by the type 2 restriction endonuclease. So first one, if you see, this is 5' TTAA CCGGAA. But if you read in the reverse direction, TTCCGG. So that is not palindromic. Similarly, 2 is also not palindromic. But 3, if you see, TTAA, this way also TTTAA, TTAAA, TTAA. So 3 is palindromic. So that will be recognized by the type 2 restriction endonuclease. So the answer here is option 3. Then next one. Which one of the following is incorrect about DNA microarray? It is used for transcriptome analysis. It is used to identify expression of microRNAs only. All the expressed genes of the sample can be analyzed simultaneously. It can be qualitatively as well as quantitatively analyze expression of a particular mRNA. So they have asked, which is incorrect about DNA microarray? It is used to identify expression of microRNA only. Not only microRNA, it can be used for mRNA also. So the answer here is option 2. Then phytoin synthase. Phytoin synthase is golden rice, Bernays Burstar system B1 DMH11 mustard hybrid, EPSPS, it is Roundup ready soybean, Cry A1C is BT cotton. So the answer here is option 3, A3 B1 C4 D2. Then before coming to the next question, I would like to tell you about what is original antigenic sin. So when a body, when it first encounters an infection, it produces effective antibodies against its dominant antigen. So these are the blue ones are the dominant antigen. This is the recessive antigen. It is present in lesser number, whereas the dominant antigen, it is present in higher number. So against this dominant antigen, antibodies will be produced and thus eliminate the infection. That is your primary response. Then after primary response, memory cells will be produced. They will remember next time infection comes, memory cells will again respond to this dominant antigen and it will be eliminated from the body. But in case of influenza viruses or coronavirus, what happens is the original antigen to which this particular primary response was elicited, that is your dominant antigen, that will become recessive and other recessive and there will be a new dominant antigen which is being created. So what will happen? The body will recognize the antigen, but it is only a recessive antigen. It is not dominant. So only but and these new dominant antigens, it will not be recognized. It will elicit only a primary response, but memory cells will not be able to recognize the new dominant antigens. They will recognize only the recessive antigen. So the immune response will not be very powerful. Okay, that is called as antigenic sin. Now given below are two statements. One is labeled as assertion A and the other is labeled as reason. Normally, the influenza virus changes its structure slightly for each passing year. Although we are being exposed to these modified or slightly mutated and evolving influenza strains every year, we do not always come down with the flu disease even if we have not received the yearly influenza vaccine. However, sometimes we do get really bad case of the flu despite the fact that we have memory cells left from the previous infection with influenza virus. Now, why does this happen? This is because of what is called as original antigenic sin, which suggests that we only mount a primary response once we have exhausted the potential to use our memory cells to eradicate the infection. Now, new dominant antigens have been produced. So once it undergoes mutation, that is this influenza virus, once it undergoes mutation, new dominant antigen will be produced. Since most of our first encounters with influenza will vary, the years in which all of the key influenza epitopes are significantly new to each of us will also vary. It is only in these years that we experience a new primary response to influenza virus, and therefore symptoms of the flu are most severe. So both A and R are true, and R is the correct explanation of A. What they have told here is that even if you get influenza disease each year, sometimes what happens is that you may get a case of the flu where you are seriously infected with influenza virus. Why does this happen? This is because influenza virus got mutated. New dominant antigen has come on its surface, and the original dominant antigen, it has become recessive. So antibodies, memory cells will recognize only the old dominant antigen, not the new dominant antigen. So that old dominant antigen, it is in the recessive condition now. So it will not elicit a primary response. Memory cells are there, they will attach there, but it will not be able to eradicate the influenza virus because new dominant antigens are present. So what will happen? Against the new dominant antigens, a primary response will be elicited again because memory cells will not be able to eradicate the infection. So symptoms of the flu will be most severe. Then next one. Xeroderma pigmentosum is an autosomal recessive disorder. It is caused due to the defects in nucleotide excision repair pathway. Answer is option 1. The cell mass of E. coli can be chemically represented as CH1.77, CO 0.49, N 0.24, the degree of reduction of the biomass. So multiply these values with the valency of the element. So carbon is only 1, so 1 into 4. Hydrogen is 1.77, so 1.77 into valency of hydrogen is 1. Oxygen valency is minus 2, so 0.49 into minus 2. Nitrogen valency is minus 3, so 0.24 into minus 3. Solve it and you will get answer as option 4, that is 4.07. Then next one. Sterilization of fermentation media with steam is performed at, so it is carried out at 121 degrees Celsius for 20 minutes. Which of the following DNA is in its double-stranded form will have the highest TM? So the one in which there is maximum number of CG or C triple bond G bonds, it will have the maximum highest TM. This is because C triple bond G is more difficult to break than A double bond T. So you have to see which one is having more content of G and C. If you see the first one, the number of G will be 1, 2, 3. C will be, okay, C and G will be 1, 2, 3, 4, 5, 6, 7, 8, 9. If you see the second, it will be 1, 2, 3, 4, 5, 6, 7, 8, 9. If you see this one, it will be 1, 2, 3, 4, 5. If you see the fourth one, it will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So the answer here is option 4 because it is having the maximum number of C and G content. Then next one. Which of the following is true for a water sample with BOD value of more than 50 ppm? So the dissolved oxygen content would be less than 6 ppm. That itself is the answer. The water is clean. The more the value of BOD, it will indicate increased amount of organic matter in the water and the water is heavily polluted. So water is clean is not correct. The amount of organic matter is very less. It is more. The aquatic life will be thriving. Aquatic life will not be thriving because it is heavily polluted. So the answer here is option 1. The next one. Alkylase. Alkylase is bioactive peptide. Lipase is for degreasing of leather hides. Beta amylase is maltose rich syrup. And phytase is animal feed. So A4, B3, C2 and D1. Answer is option 4. So next question. A monoclonal antibody was heterologously expressed in E. coli and Chinese hamster ovary cell line, and the recombinant protein obtained were purified to homogeneity and were found to be structurally identical. However, the protein expressed in E. coli was not functionally active. The reason could be, so they have taken a monoclonal antibody from another source and they have expressed it in E. coli as well as in Chinese hamster ovary cell lines. Then the recombinant protein which was produced was extracted and purified and was found to be structurally identical. That is, the protein extracted from E. coli and Chinese hamster ovary cell line were found to be structurally identical. But the protein expressed in E. coli was not functionally active. The reason could be, the reason for this is that glycosylation does not take place in E. coli, whereas it takes place in Chinese hamster ovary cell line because it is a eukaryotic mammalian cell line. E. coli is prokaryotic, so glycosylation doesn't take place in E. coli. So the protein needs glycosylation for its function. That is the answer. The next one. Which of the following factors do not affect the operation of a trickling filter? Organic loading, hydraulic flow rates, temperature of the water and air, linear flow velocity of the liquid. So which does not affect the operation of a trickling filter is linear flow velocity of the liquid. Answer is option 4. Next one. The pH indicator for animal cell culture media is, it is phenol red. Answer is option 2. Match list 1 with list 2. List 1. Coenzyme A. Coenzyme A is pantothenic acid. FAD. FAD is flavin adenine ribonucleotide, so it must be riboflavin. FAD is B1. Then thymine pyrophosphate. Thymine pyrophosphate is vitamin B1. Thiamine is vitamin B1. NAD. NAD is niacin. D3. So the answer here is A2, B1, C4 and D3. So answer is option 2. Then next one. Fermentation of fruit juices to vinegar involves use of yeast as well as acetic acid bacteria. Answer is option 4. The electron transport system is present in inner mitochondrial membrane. That was a very simple and a direct question. Okay, next one. Which of the following groups of viruses generally infect plants? It is riboviruses that generally infect plants. Riboviruses. Answer is option 2. Then, the issue of pesticide contamination of carbonated drinks was brought into limelight by which of the following NGO? That is Center for Science and Environment. Answer is option 1. Find the odd one out with respect to the pleiotropic gene expression. So pleiotropism is the condition in which single gene controls more than one phenotypic effect. So white eye gene in Drosophila, flower color gene in Pisum sativum, Marfan's syndrome, plumage color in poultry. So white eye gene in Drosophila is recessive X-linked character. Marfan syndrome is affect the connective tissues and it is an autosomal dominant disorder. It is caused by mutation of the FBN1 gene that makes fibrillin, which result in abnormal connective tissue. Then plumage color in poultry. So they have asked for the odd one out, that is flower color gene in Pisum sativum. Answer is option 2. Then the addition of silver nitrate in plant tissue culture media is useful for managing and minimizing detrimental effects of which of the following plant hormones? So silver nitrate, it inhibits the action of ethylene. So answer is option 2. Then, in a typical bacterial growth curve, the phase in which cells adapt to growth condition and are metabolically active but not able to divide is. So, the lag phase is generally characterized by metabolic activity but not growth. The cells utilize the media and synthesize small molecules that are necessary for replication. So, the answer here is option 2. Exponential phase is time of exponential growth or the log phase of growth. Then stationary phase is the growth reaches a plateau as the number of dying cells will be equal to the number of dividing cells. So the answer here is lag phase. Then next one. Two statements are given. Statement 1. Protein ligand docking involves binding of a small molecule to the protein receptor. Yes. Ligand is bound to the receptor that is involved in protein ligand logging. Both Autodock and Ludi1 are protein ligand docking programs. That is also correct. So both the statements 1 and 2 are true. Then next one. Which of the following is not correct about mutagenesis using chemical mutagen? Low frequency of mutation in the gene of interest. That is correct. Inability to confirm the location of the causal mutation even after getting the desired phenotype. So that is correct. Then creation of point mutation at a specific position in the gene of interest. That is not possible. So they have asked which is not correct about the mutagenesis using chemical mutagen. So answer is option 3. Then which of the following bacterium is generally used in plant genetic engineering? It is Agrobacterium tumefaciens that is usually used. The microglial cells found in human brain represent which of the class of innate immune cells? So they are the macrophages. Answer is option 2. Which of the following is an enveloped virus? So it is dengue virus. Answer is option 1. Vaccination in the last century had resulted in 100% reduction in active cases across the globe. 4. So that is for the case of smallpox. There was 100% reduction in the active cases. So that is option 1. The study of various atmospheric parameters is called. It is called meteorology. Meteorology. Answer is option 3. Geology is study of earth. Hydrology is about water. And environmental biology everybody knows. So meteorology is the answer. Then next one. A culture of E. coli cells was diluted 10 raised to 6 fold. So dilution factor is 10 raised to 6. And 100 microliter of this was used for plating. After 24 hours of incubation, the number of colony forming units was found to be 180. Sorry. The CFU count of the original culture is, that is number CFU into dilution factor divided by volume plated. So volume plated they have given as 100 microliter. Convert it into ml, you get 10 raised to minus 1 ml. Now substitute 180 into 10 raised to 6 divided by 10 raised to minus 1, you get 1.8 into 10 raised to 9 CFU per ml. Coming to the next question. Given below are two statements. Statement 1. Orthologs are sequences from different organisms or genomes derived from speciation events, while paralogs are sequences from the same organism or genome which are derived from gene duplication event rather than speciation events. So first statement is correct. Orthologs are sequences from different organisms derived through speciation. Paralogs are sequences from the same organism derived by gene duplication events. In the light of the above statement, choose the correct option from the option given below. Statement 2. Both orthologs and paralogs are homologous sequences. So both the statements 1 and 2 are correct. Answer is option 1. Then next one. Which of the following is structurally independent three-dimensional unit associated with a particular functional role? It is called the domain. Answer is option 1. Next one. Which of the following is multinucleated cell present in the bones of human body? They are called as the osteoclast. Answer is option 2. Then which of the statement is not correct with respect to cybrids? They are the resultant of spontaneous fusion of protoplast obtained from common callus culture. That is not correct. So first statement itself is not correct. They allow the direct transfer of cytoplasmic male sterility from donor to recipient. That is also correct. The process can bypass up to 12 back crosses required in the development of alloplasmic lines. That is correct. It is a fusion of protoplast of one parent and cytoplast of the second. That is correct. Not the fusion of protoplast obtained from common callus culture. It is not the fusion of protoplast. It is fusion of protoplast of one parent and cytoplast of the second will result in hybrid. So option 1 itself is not the correct answer. Then next one. The approximate length of the open reading frame coding for a 55 kDa bacterial protein will be how many nucleotide long? So 55 kDa is 55,000 Dalton. 3 nucleotides are code for 1 amino acid. And 1 amino acid is 110 Daltons in weight. So 3 nucleotides code for 110 Dalton. And 55,000 Daltons will have how many nucleotides? It will have 1,500 nucleotide. So the answer here is option 3. Then next one. Which of the following is generally not a primary metabolite? Amino acid, polysaccharide, antibiotic, vitamin. Antibiotics are not primary metabolite, they are secondary metabolites. Then next one. How much protein will be needed to make 100 microliter of 15 micromolar solution of a 95 kDa protein? So 95 kDa is 95 into 10 raised to 3 grams per liter. Molarity is given by Wb by Mb into where weight, W is the weight, M is the molecular weight, and into volume in liters. So we have to find out how much protein. So we have to find out the weight of the protein. That is Wb we have to find out. That is M into Mb into V. Substitute the values and you will get it as 0.1425 mg. That is answer is option 1. Then harmful gases such as sulfur dioxide can be removed from the industrial exhaust using scrubbers. Answer is option 4. Which of the following mineral elements play an important role in biological nitrogen fixation? It is molybdenum because nitrogenase is having molybdenum. Nitrogenase is involved in biological nitrogen fixation. Then next one. The degree of inhibition of an enzyme catalyzed reaction by an inhibitor is independent of substrate concentration. This is expected in the case of, it is expected in the case of non-competitive inhibition because non-competitive inhibition is characterized by inhibitor binding to the allosteric site and not the active site. That is active site where the substrate binds. So it is independent of the substrate concentration. So answer is option 3. Then next one. Which of the following viruses is likely to have highest genomic mutation rate? It is poliovirus. Why? Because it is an RNA virus and RNA viruses has highest mutation rates. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you. I have uploaded previous year's GAT B question papers also, like 2020 and 21 I have uploaded. So you can go through them also. It is available in my channel.